Are there key gene mutations in T-cell lymphoma? Imagine a complex puzzle where tiny pieces called genes come together to shape how cells grow and behave. In T-cell lymphoma, a type of blood cancer that affects the immune system's T-cells, certain gene mutations are like critical pieces that help the disease develop and progress. These mutations are not random. They often involve genes that control how T-cells signal each other, how they grow, and how their DNA is modified. Research shows that across different types of T-cell lymphoma, about 20 genes frequently carry mutations. These include diseases like peripheral T-cell lymphoma, angioimmunoblastic T-cell lymphoma, and cutaneous T-cell lymphoma. Many of these mutations lead to increased activity in pathways that tell T-cells to grow and divide, which can cause the cells to become cancerous. For example, one well-known mutation is in the RHOA gene, specifically the G17V mutation. This mutation is unusual because it actually reduces the normal activity of RHOA but still promotes abnormal T-cell behavior, especially in angioimmunoblastic T-cell lymphoma and peripheral T-cell lymphoma. Another important mutation involves the TP53 gene, which normally acts as a tumor suppressor. When TP53 is mutated, its ability to prevent uncontrolled cell growth is compromised, allowing the lymphoma to advance more easily. In cutaneous T-cell lymphoma, mutations in the PLCG1 gene are common. This gene plays a role in T-cell receptor signaling, and mutations here can make T-cells more prone to grow uncontrollably. Some mutations involve fusion genes, like the CTLA4 and CD28 fusion, which are often found in Sézary syndrome, a type of cutaneous T-cell lymphoma. These fusion genes cause abnormal signals that encourage T-cells to multiply and survive longer than they should. Additionally, mutations in genes that regulate epigenetic changes, such as EP300, are frequent in certain T-cell lymphomas. These changes affect how genes are turned on or off, and their disruption can lead to resistance to treatment and disease progression. Many of these mutations also impact T-cell receptor signaling pathways, which are essential for normal immune responses. When these pathways are altered, they can cause T-cells to grow excessively and evade normal controls, fueling the cancer. The genetic landscape of T-cell lymphoma is quite complex, with many rearrangements and mutations that reflected its diverse nature. Understanding these key gene mutations is vital because they help scientists develop targeted therapies that can interfere with these abnormal signals. While research continues to uncover more about these genetic changes, it is clear that mutations affecting T-cell signaling, tumor suppression, and epigenetics are at the heart of T-cell lymphoma. If you or someone you know is dealing with this disease, always consult healthcare professionals who can interpret these genetic findings in the context of personalized treatment options.